Hi, my name is Jenna.
Hi, I'm Jacquelyn.
And hi, I'm Taya. Today we'll be presenting on the RUNX1 gene and how it affects acute myeloid leukemia. A little overview of what we'll be going over today is what acute myeloid leukemia is, how the RUNX1 gene and RUNX family are significant to the prognosis of AML, explain what RUNX1 ETO is, some mutations associated with RUNX1 and how they relate to AML, different forms of treatment and pain prevention, and why certain therapies are used for different subtypes of AML, and what AML looks like in the future for us. So, a little background on acute myeloid leukemia. It's defined as a heterogeneous clonal disorder characterized by the immature myeloid and bone marrow proliferation. 19,520 people are diagnosed with AML each year in the United States. And unfortunately, there's an estimated 10,600 people that die from acute myeloid leukemia every year, just because of how aggressive the cancer is and how difficult it is to treat. Some diagnoses of AML are reliant on the morphological identification of leukemic myoblasts and their peripheral blood and bone marrow stains. Some distinguishing characteristics of the cells are their round to irregular nuclei, distinct nucleoli, and very little cytoplasm. In order for someone to be diagnosed with leukemia, they have to have over 20% leukemic blasts present in their bone marrow and in their blood. AML is a very aggressive cancer, like I said before, and the prognosis of it in general is not good, but when RUNX1 is present, it's even worse. Next slide. Thank you. This is a depiction of the development of different types of blood cells and stuff like that. So myeloid stem cells and lymphoid stem cells are depicted here. Myeloid stem cells develop into red blood cells, myoblasts, and platelets. Myoblasts then differentiate into granulocytes, which then become basophil, eosinophil, and neutrophil cells. These are all forms of white blood cells. The lymphoblasts develop into B lymphocytes, T lymphocytes, and natural killer cells, which are also classified as white blood cells. As I mentioned before, with the diagnosis part of AML, if any of these blood cells obtain any abnormalities, it's likely that the patient will have acute myeloid leukemia. So for a little background on RUNX1, its function is it's a main transcription factor that is essential for initiating hematopoietic differentiation. It signals the differentiation of myeloid blood cells, B cells, and T cells, which are crucial for the maturation of blood cells. As for structure, it belonging to the RUNX gene family, the structure of RUNX1 protein is characterized by the N-terminal RUNT domain. RUNX genes are responsible for the mediation of DNA binding and interactions with the heterodimeric core binding factor. The core binding factor subunit beta is a protein that in humans is encoded with CBFB gene. The protein encoded by this gene of the beta subunit is a heterodimeric core binding transcription factor that belongs to the PEPB2 CBF transcription factor family, which is a master regulates a host of genes that are specific to hematopoiesis. The RUNX1 specifically is the non-binding factor of core binding factor beta. in the C-terminal transcription domain. When RUNX1 and core binding factor produce a heterodimeric structure, the affinity between RUNX1 and DNA increases. This is because the core binding factor beta promotes a DNA recognition loops to form, which increases the affinity between DNA and RUNX1. And as I mentioned before, the presence of RUNX1 mutations has been proven to have a very large negative impact on patients with AML.
Like Taya just said, RUNX1 mutations can lead to AML or other pre-leukemic diseases. And RUNX1 mutations that directly lead to AML are most likely due to the decrease in functionality of wild-type RUNX1. Without the presence of wild-type RUNX1, immature and non-functional blood cells accumulate, and over time, there's a decrease in hematopoietic differentiation without the wild-type RUNX1. Both of these factors together lead to the development of leukemia. Without the wild-type RUNX1, studies have shown that the amount of core binding factor beta also decreases. And in its normal state, core binding factor beta increases the binding affinity between RUNX1 and DNA. Therefore, when core binding factor beta proteins decrease, the binding affinity between RUNX1 and DNA also decreases. This will ultimately lead to the non-regulation of hematopoiesis and transcription. So there are many mutations that can occur with RUNX1. However, the mutation that results in AML is the translocation between chromosomes 8 and 21. This is a chromosomal mutation, and it involves the translocation of intron 5 on RUNX1 and intron 1 of ETO. This produces the RUNX1 ETO complex, and ETO is a myeloid transforming gene. And during translocation, it provides 4 nervy homologous regions to RUNX1. These regions can act as repressors or co-repressors that are involved in gene sequencing. Therefore, it further promotes the increasing rate of mutations. So this picture right here just shows the 4 nervy homologous regions that ETO provides for the RUNX1. Together, it creates a RUNX1 ETO complex. So the RUNX1 ETO complex produces an oncoprotein, and oncoproteins causes modifications in healthy cells and transform them into tumor cells. With this being said, the RUNX1 ETO complex is a big reason for onset leukemia. The complex is also responsible for decreasing the expression of genes found in base excision repair. For example, the enzyme OOG1 is responsible for catalyzing the ligation of guanine nucleotides that are oxidized. However, with the presence of RUNX1 ETO, cells have been seen to have a decrease in expression of this enzyme. Therefore, this is an issue because the guanine nucleotides have been exposed to reactive oxidative species. Another example is the BRCA2 and ATM regulators. These are 2 common regulators found in homologous recombination, and cells expressing RUNX1 ETO have also been seen to decrease the expression of these 2 regulators. And this is a big issue because without these genes, the repair mechanisms will not work properly, and more mutations and more damage will continue to accumulate. Aside from RUNX1 ETO leading to onset leukemia, this is not the only mutation that occurs. There are actually many mutations that co-occur with the RUNX1 mutation, and the RUNX1 mutation only increases the chances of AML, but it does not cause AML. This is why the two-class model has been developed to analyze the effect of multiple mutations. Mutations that are hyperactive in increasing cell survival and rapid cell growth belongs to class 1, whereas class 2 are mutations that aren't as hyperactive, but promote the decrease in hematopoietic differentiation. RUNX1 is actually a class 2 mutation, and in older patients, we see RUNX1 co-occurring with ASXL1. ASXL1 is a class 1 mutation, and it's a gene that codes for chromatin rearrangement. So the co-occurrence of RUNX1 and ASXL1 results in the lack of response in therapeutic treatments. In younger patients, we see RUNX1 co-occurring with ROS or RAS. RAS is a class 1 mutation, and together ASXL1 and RAS are accountable for abnormal cell proliferation, and we see an increase in kinase activity and decrease in RNA splicing, which leads to the increase of leukemic activity when there are both class 1 and class 2 mutations together.
Okay, so RUNX1 has always been thought to only affect hematopoietic systems, as Jacquelyn and Taya have mentioned, but it can also be found in other cancers such as hormonal cancers and skin cancer. So in breast cancer, it affects the basal and luminal compartments of the mammary ducts. There's also a hypothesis that RUNX1 is actually a tumor suppressor. So it suppresses the epithelio to mesenchymal transition process because RUNX1 isn't as prevalent in comparison to other tumors. But there's also a strong correlation between RUNX1 mRNA levels and a poor prognosis in triple negative breast cancers. RUNX1 has been found to amplify a portion of neuroendocrine castration-resistant prostate cancer, but it's not as common for men to possess RUNX1 as women. So along with the hormonal cancers, as I mentioned, it's also found in skin cancer. So the loss of RUNX1 decreases the amount of human oral skin squamous carcinoma cells, but RUNX1 is actually very important for the survival of keratinocytes in skin. So overall, it's very important to observe RUNX1 in other cancers because we need to be able to target this mutation. Next. So along with the mutations that Jackie mentioned, there's also TP53 mutations, which is why chemotherapy doesn't really work for AML. It is a very common and well-known cancer treatment, however, but the TP53 mutations cause chemo resistance. So for patients, next slide, sorry. So patients who are RUNX1 ETO positive, chemotherapy will actually target the oncoprotein, but because there are serious health consequences and it requires a high dosage to achieve any sort of progress, many elderly patients and younger children do not qualify for this treatment. Can you go back? Yeah, sorry. So there are many treatments that are still in works being approved. So there's immunotherapies, genetically targeted therapies, non-genetically targeted therapies, and drugs that are still going through clinical trials. A promising genetically targeted therapy is by administering decitabine with the standard 7+3 chemotherapy. It targets the TP53 mutations, and if it's used consistently, then it can prevent the relapse of the cancer. An immunotherapy that is promising is the natural killer cell-based immunotherapy. What it essentially does is it will be able to kill leukemia through the graft versus leukemia effect. It can promote engraftment from T cells, and it will protect against graft versus host disease. Next.
So this is another treatment that we found. As mentioned before, the RUNX1 and the co-binding factor beta form a heterodimer for DNA binding and regulation of gene expression. Mutations in this protein complex have been proven to cause AML. This new treatment involving an inhibitor called Rho53335 directly interacts with the RUNX1 and core binding factor beta to prevent these mutations from happening. So in that first row, you can see the normal interaction between core binding factor beta and RUNX1. And then on that second line, you see the Rho53335 inhibiting the interactions between the RUNX1 and the core binding factor beta. And so those mutations can't even occur because the binding doesn't even happen. This has been successful in some zebrafish and other sorts of model organisms that have been proven to be good with AML trials.
So the future of AML is still very unknown, as AML is a very invasive and aggressive cancer. Chemotherapy doesn't work too well, but there are still treatments, like I mentioned, being worked on. However, treatments in regard to RUNX1 should be researched more, as that is the primary gene or primary mutation that affects AML. Research from other types of cancers that have RUNX1 can be used to determine how to fix this. And overall, in the end, we just want to increase the survival rate and prognosis for patients who have AML. And references.
That is all.